time we have to move on. The
Thank
next
you.
speaker, thank you. The next speaker is Jonathan Ma from the University of Washington.
I think my mic is on. Okay. Thank you, Professor Ogbuno, for the introduction, and also thanks to the organizers for giving me this chance to share my work. Anyways, my name is Jonathan Ma. I am an undergraduate working with Dr. Jesse Bloom's group at the University of Washington and the Fred Hutchinson Cancer Research Center. And today, I'll be discussing some of my ongoing work concerning methods for identifying sites under diversifying or positive selection on viral proteins. So an important goal in genetics is to identify which mutations lead to evolutionarily significant changes in phenotype. I study this through the lens of protein evolution, and specifically, I am interested in identifying positively selected for sites which alter antigenicity in viral proteins. As sort of a cartoon example, here on the left, we have a purple virus bound by a blue antibody. However, because virus evolution is both extremely rapid and driven by antigenic pressures, very quickly, we find that after a few mutations, which I've shown here as a color change, but more realistically would be a change in structure conformation, the blue antibody is no longer able to bind to the now yellow virus. Unfortunately, this problem is very rarely that straightforward because not all mutations that fix do so because they confer some sort of beneficial phenotypical change. Rather, some mutations fix due to other forces, such as genetic drift, in which a small population subject to bottlenecks loses certain genotypes and phenotypes, or genetic hitchhiking, in which neutral or even deleterious alleles can become fixed due to their physical proximity to sites which are positively selected for. And while these are very popular examples in population genetics, at this point I should now disclose that we are approaching this problem via a phylogenetics framework, which comes with its own host of problems and assumptions. So in phylogenetics, the basic strategy for identifying any sort of biologically interesting selection is to first define a null expectation for protein evolution in the absence of non-purifying selection. Then, supposing that the null expectation adequately describes evolution, any sites which deviate from the model are identified as sites where mutations have been selected for or against, specifically because they alter evolutionarily relevant phenotypes. So a traditional null model is to assume that non-synonymous and synonymous mutations fix at the same rate. That is to say, their ratio, denoted here as D and DS, or parameterized as omega, is assumed to equal 1 for neutrally selected sites. Then, sites with DNDS greater than 1 are under positive selection, while sites with DNDS less than 1 are under negative selection. And something I would like to really briefly note here is that in most subfields of evolutionary genetics, when we consider DNDS, we do so over a large number of sites. However, for viruses, because they evolve so rapidly, we're actually able to get meaningful information from looking at DNDS and omega on a site-specific basis. Again, as a very simple example, if we take some hypothetical genome with a mean substitution rate of 4 synonymous mutations per site per X amount of time, then this site here on the left, which has 8 non-synonymous mutations, would be considered under positive selection, while this site here on the right, which has 3 non-synonymous mutations, would be considered under negative selection. And this DNDS null model for evolutionary rate has, certainly its strengths in that it is intuitive and straightforward and easy to work with. However, it can often be misleading. So a key assumption of this traditional null model is that the evolutionary properties of sites are independent and identically distributed. However, one of the problems with this assumption is that sites are not, in fact, identically distributed. For example, in protein folding, cysteine bridges are typically thought of as very important. So the sites near these cysteine bridges, disulfide bridges, are probably really heavily conserved. So to address this biological reality that sites do not have identical evolutionary properties, we evaluate protein evolution on a site-specific basis. We do this by making use of empirical data from a high-throughput functional assay known as deep mutational scanning, or DMS. In summary, we take a reference protein, use reverse genetics encoded plasmids and PCR mutagenesis to create a library of all possible single codon variants, passage this library through some form of functional selection, and then use deep sequencing to measure the relative abundance of each codon variant before and after selection. In the case of viral proteins, this functional selection is growth in cell culture, which importantly does not include antigenic selection. Then, single codon variants which are enriched are assumed to correlate with mutations that are biophysically favorable, while the opposite is true for variants that are depleted. And we visualize the results of this assay as a logo plot, describing the amino acid preferences of each site, in which the height of a letter corresponds to the relative abundance of that amino acid at that site. Let's now revisit that example from earlier, but this time with a null model informed by our experimental data. We now see that at certain sites, such as this one here on the left, there's a high degree of amino acid tolerance, meaning that we observe several different amino acids at about the same prevalence. So in a sense, our null expectation is able to account for the observed result of there being a high number of non-synonymous mutations, because at this site, evolution is basically a non-biased random walk. However, if we look again at this site here on the right, we now see that these 3 non-synonymous mutations occurred at a site which is heavily conserved to be either a glutamic acid or a glutamine. And notably, these 2 amino acids are very closely related in terms of biochemistry. So our null expectation suggests that this site is very strongly conserved. For there to have been any sort of non-synonymous mutations on the site on the right is actually quite surprising. And we think is actually stronger evidence of positive selection than here on the left, even though the site on the right had a D-index ratio which was less than 1. So in terms of numerical implementation, our null expectation, denoted here as P, is a codon selection model given by the product of 2 transition matrices, mutation, denoted as Q, and fixation, denoted as F. We parameterize our map of amino acids, acid preferences as pi at site R for all DNA relevant amino acids. And as a short summary, Q is an HKY85 codon selection model, which basically helps differentiate between transitions and transversions, while F contains several important terms, of which I've highlighted 2. In blue here on the left, we have omega, which is again the estimate of evolutionary rate, while in red here on the right is pi, which is our empirical data. And this whole model, which is known as an experimentally informed codon model, or EXPCM, has previously been shown to have a better phylogenetic fit than other null models for protein evolution. From here, we can compare the expectation from our null model to observed evolutionary histories from natural sequence alignments. Importantly, since the functional selection component of our assay does not include antigenic selection, any discrepancies between expectation and observation can be attributed to mutations selected for or against because they affect antigenicity. And there are 3 sort of overall approaches for this comparison, but I will be only describing 2. So in a fixed effects likelihood approach, you individually estimate a value of omega for each site. But the drawback of this approach is that for larger proteins, such as influenza hemagglutinin, which has 565 sites, you need to make 565 different and independent estimates of omega. And as a result, you need a very stringent cutoff for statistically significance to satisfy the restraints for multiple hypothesis testing. And this stringency tends to lead to a high number of false negatives. To get around this issue, we decided to use a random effects likelihood approach, where we instead fit a gamma distribution describing omega across the entire gene. We then partition this gamma distribution into several discrete categories. Here I have shown 4, but realistically it's more around 100. And then assign each category the mean omega value of its subdistribution. And the reason why we're going forth with the random effects likelihood approach is that this really only has 2 hypotheses, which are the shape and scale parameters of our gamma distribution. Once we have a partitioned gamma distribution of omega values, we can approximate its integration using an empirical Bayesian approach. For the theoretical folks, this is just a brief overview, but a more intuitive explanation is that we are computing the posterior probability of observing a particular omega for a given site, corrected by the probability of observing any omega at that site with respect to our distribution. As a short example, let's revisit that same example gamma distribution from the previous slide. If we were to choose some sort of cutoff, like omega equals 1, we can then take the sum total probability of observing any category whose omega is greater than our cutoff, which again is 1, and this gives us the posterior probability of observing positive selection at that site. So to evaluate the statistical power of our approach and compare it to others, we performed a simulation study in which we randomly selected 3% of sites and simulated them as being under positive selection with varying levels of omega, 5, 10, 15, 20, 25, and 30, while keeping all of their sites as simulated under neutral selection. We then applied our EXPCM-based approach, which is shown in red, as well as the YNGKP approach, which is shown in blue. And to clarify, the YNGKP approach is just a very commonly used site uniform codon selection model. We did this whole thing with 5 independent sets of randomly selected sites, and then plotted out the average true positive and false positive rate to show you these here ROC curves. And something I'd just like to really quickly point out is that For almost all levels of omega, the red curve tends to perform better than the blue curve, and especially so at higher levels of omega. So we've begun applying this method towards identifying sites on actual viral proteins. Here I have shown the 3D structure of 2 strains of influenza hemagglutinin, color-coded with sites under varying levels of positive selection. One thing I would like to point out is that the vast majority of these sites under positive selection are located on the head region of the protein, which is consistent with our knowledge of antibody binding sites, providing evidence that we are in fact detecting signatures of antigenic selection. So to briefly review our findings, simulations show that our site-specific null model and random effects likelihood approach outperform current methods for identifying sites under positive selection. When we apply this to real data, our findings are consistent with what we know and expect from antigenically important viral proteins. In the future, we'll be applying this to Zika virus envelope protein and HIV envelope glycoprotein, as well as possibly other medically important viral proteins. Somebody told me that there happens to be some sort of like global pandemic going on, so that sounds pretty useful. And ultimately, we hope that our approach and results can help inform preventative measures like vaccine strain selection, as well as identify possible targets for structure-based therapeutics. As always, thanks to my super awesome mentors, Sarah Hilton and Jesse Bloom, as well as all of the wonderful people in the Bloom, Lohmiller, and Grid labs for their guidance and support. And additionally, huge thanks to several groups of also super awesome people. This work was funded in part by financial support from the University of Washington Departments of Microbiology and Genome Sciences, the Mary Gates Endowment, and the Washington Research Foundation. Otherwise, I'd be happy to take any questions if there are any. And if I don't get to you, please feel free to reach out to me via this contact information. This is what I look like, by the way. All right.
Great. We have time for a couple of questions. First one from Kelsey Byers. What happens with lower, more typical levels of omega comparing between XPCM and YNGKP?
So I assume you mean if we were to simulate, or basically do the same simulation study, but with an omega that's of lower value, like maybe like 1.5 or something. I would expect that in a model comparison test or a likelihood comparison test, our model would still perform better, but we haven't done that specific simulation, so I can't definitively say that the ROC curve would be better.
Great. One more question, a short one it seems, from Lee. Hi, Jonathan. Will your approach be able to apply to the other organisms rather than only viruses?
Absolutely, yes. The crux of the assay is that it has to be a protein. So actually, we do have people doing this for like our antibodies grown in yeast or something. And we have also published a study that looked at, I think, REC-A in E. coli. So absolutely, this protein-specific approach can be applied to any protein.
And once again, to everyone else, you feel free to kind of still write your questions in the Q&A. They will be forwarded to the speakers at a later date. In the interest of time, we will move on.